This video shows how to passage iPS and ES cells cultured in StemFit medium. The passage methods of iPS-ES cells are roughly classified into clump method and single cell method. This video is focusing on the single cell method with StemFit medium. In the clump method, a non-enzymatic dissociation reagent is used to make clumps. In the single cell method, an enzymatic dissociation reagent is used to make colonies into a single cell. In the clump method, clumps are diluted at a rate of about 3 to 50 times and seeded, whereas in the single cell method, cells are counted and seeded at a density of 1,000 to 5,000 cells per centimeter squared. In the single cell method, it is necessary to add ROCK inhibitor for 24 hours after passage in order to suppress cell death. "24 hours after passage, ROCK inhibitor is removed. At day 7, cells are passaged again. It is recommended that cultures switch from the other media to StemFit medium 2 days before the passage for adaptation of cells. To change medium in 6-well plates, add 1.5 ml of the prepared StemFit medium into each well. Gently add medium to each well by placing the pipette on the edge of the well. Use StemFit medium at room temperature without heating. The following instructions are for the passage of 1 well of a 6-well plate. Transfer 4 ml of the prepared StemFit medium to a new 50 ml tube to prepare the passage medium. Add the ROCK inhibitor Y27632 to a final concentration of 10 micromolar. Hereinafter, this medium is referred to as the passage medium. Mix well by pipetting. The remaining medium can be stored at 2 to 8 degrees for up to 2 weeks. Take out the culture plate from the incubator and observe the cells under a microscope. Observe cell density, colony size, and cell morphology to confirm that the cells are suitable for passage. Record pictures of the cells as needed. Remove the medium using an aspirator. Gently add 1 ml of PBS to the well by placing the pipette on the edge of the well. Remove PBS immediately. Gently add 500 microliter of cell dissociation reagent and spread it over the whole well. Return the plate into the incubator and incubate at 37 degrees for 5 to 10 minutes. The dissociation condition needs to be optimized for the cell line. Observe the state of cell dissociation with a microscope. Confirm that cell-to-cell adhesion is loose. If dissociation is inadequate, return the plate into the incubator and extend the incubation time. Detach cells by pipetting up and down with a 1 ml pipetter. Pipette gently to avoid foaming. Transfer cells into a new plastic tube. Add 500 microliter of passage medium to the tube containing the cell suspension. Collect cells by centrifugation at 300 times G for 5 minutes at RT. Aspirate the supernatant and resuspend cells in passage medium. Carefully pipette cells up and down about 10 times using micropipette to obtain a single cell suspension. Gently pipetting to avoid foaming. Cells can be left in the tube for about 1 hour, although immediate seeding is recommended. Take a portion of the cell suspension and count the viable cells. Mix cells with a tri-band blue and apply to cell counter. Observe cells under the microscope to ensure they are dissociated into single cells. Count living cells and determine the cell concentration. Calculate the seeding number. Adjust the seeding cell number depending on cell lines and condition of the cells. Prepare matrix coated plate. Refer to the product instruction for a coating process. Remove coating solution from a well and add 1.5 ml of passage medium immediately. Solution should be prepared quickly, not to allow the coated wells to dry out. Mix cell suspension with gently pipetting up and down. Seed the appropriate number of cells to a well. Immediately agitate the plate vertically and horizontally, taking care not to spill the medium. Observe the cells under a microscope to ensure that cells evenly distribute over the well. After seeding, place the plate into the incubator immediately and keep it warm. Use passage medium containing rock inhibitor for passaging. Avoid additional freeze-thaw cycles. Store medium in 2 to 8 degrees for up to 2 weeks. Wash cells with PBS gently and quickly. Optimize the incubation time of cell dissociation depending on matrix and dissociation reagents. Optimize seeding cell number depending on the cell line. Change medium the next day with StemFit medium without rock inhibitor. Do not disturb the plate for 24 hours. This is the recommended schedule for medium change. It is recommended to passage the cells every 7 days. According to this schedule, medium change on weekend can be skipped. This time-lapse image shows colony morphology changes. Passage cells when cells reach 60 to 80% confluency.